Good morning, everybody. So I now notice sitting up here, I can't see anything, so I assume that the hall is completely packed for this really exciting session. We would like to, my name is Esther Sammler, I'm from the University of Dundee, and we will be introducing our co-chairs and co-speakers in a second. But we first would like to somehow thank Parkinson's UK, because this session on endolysosomal function and lipid metabolism in Parkinson's disease is sponsored by Parkinson's UK. So my co-chair is Tony Shapira, whom probably everybody will know, and then Emil Gustafsson and Verena Fanning from, I think like all of us have a connection to the UK, but you're the only one at the moment coming from the US. So thank you so much for coming. So we would like to make this session really interactive. We have 4 talks, and the talks are 20 minutes long, and we will try to be on time, and then there will be 5 minutes for questions. And I would really invite everybody to participate in the discussion, because that's what really makes it fun. So our first speaker is Emil Gustafsson. He is well known to everybody, so he's a geneticist working at UCL and also with association with the Dementia Research Institute, working with Mina Reiten. And Emil will talk to us about cryptic transcripts in Parkinson's disease. So with no further ado, Emil, please go ahead.
Well, thank you for that introduction, and I want to thank the organizers, and particularly Parkinson UK, for allowing me to come here and present my work today. So I have a quite general title here, but I'm stretching it a little bit. What I'm going to talk about mainly is how we kind of have to go away from this gene-centric view of looking at function and impact of variants, and actually start looking at the transcript as the unit. So these are my disclosures. So as I said, in general, I'm going to do a brief introduction about Parkinson's and why it's important to understand transcript diversity. And I'm going to exemplify this by looking at GBA1 and its pseudogene GBAP1. And lastly, I'm going to talk a bit about how actually using transcriptomics can improve therapeutic designs. And I'm going to do that with the synuclein as the example. So Parkinson's disease is largely a movement disorder, although there's a lot of non-motor symptoms as well. It is characterized by this loss of dopaminergic neurons in the substantia nigra. You can see them in the black here. And the hallmark Lewy body pathology, which is aggregated synuclein in fibrillar or aggregated structures. So Parkinson's disease is a progressive disorder in that at the time of diagnosis it's largely the movement symptoms, but it really starts 1 to 2 decades before that with things like constipation, REM sleep behavior disorders. And after the onset of movement disorder, there's also a lot of non-motor symptoms such as dementia that's actually prevalent. And while this is not really exactly how this is actually progressing, but it is also progressive accumulation of this pathology, the synuclein I said, that then starts in the vagus nerve, goes to midbrain, and then cortical structures. So why not completely? Parkinson's disease is a genetic disorder, and Christine Klein was presenting this yesterday at the plenary talk, where you have things like synuclein and VPS35, which are these quite penetrant variants with a large effect size, but they are rare. Then we have things like RAB32, which we just recently discovered, and LRRK2, which are a bit more common and have a slightly low effect size. And probably the most important one of them all is GBA, which I put here. And then we have everything that we see in the complex forms or the sporadic forms of the disease, which are these GVAS hits. But when you start looking at this together, there's actually certain pathways, there's certain biological functions that start really becoming important. I'm going to highlight them, but obviously in the title as well, is this endolysosomal function. You can see here the monogenic genes, and you have some of the risk genes as well here. And even when you do this at a burden analysis, you see the same thing. The lysosomes, the mitochondria comes up. And again, I have to emphasize GBA, which is probably the most important risk factor for Parkinson's disease. But before that, I just want to introduce this concept of, well, why we need to understand transcripts. So you can look at transcripts as a map of your genome or your transcriptome, where each gene then generates multiple transcripts, and these can sometimes have different and even antagonistic functions. They can be expressed by cell types and tissue. And they're obviously important then to first understand the gene function, but also to appropriately interpret variants, but also for diagnosis. And lastly, which I think is extremely exciting, is for the design of gene or RNA-based therapies. And I'm going to touch a bit on that in the end. So just briefly to go over this, the transcriptome annotation, as I said, is incomplete. And I think the latest version of GenCode, they doubled the number of transcripts. And this is because of the implementation of things like long-read RNA sequencing. So there's about 250,000 transcripts we know across the genome. This annotation is incomplete, and it's not always the same. So you can see here, the highlight on this is that it's particularly in brain where we see this, because the transcriptome complexity is higher in brain than any other tissue. We also know that this is by cell types, and I just highlighted in red here, these are oligodendrocytes, and it's probably because it's a very poorly studied cell type in the brain. But also by genomic regions, we have complex regions, repetitive regions, or highly homologous sequences. So bring it back to GBA, as I mentioned previously. So GBA then encodes glucocerebrosidase, where biallelic mutations cause Gaucher disease, a lysosomal storage disorder, whereas heterozygous variants or mutations are probably the most important risk factors for Parkinson's disease. And as I said, when you have then mutations in GBA, you have this dysfunction is this hydrolase of glucoceramide, and you see this lysosomal accumulation of the substrate in the cells. So why but beyond this being a very important risk factor, GBA also, something happened down here. GBA has, as I said, a pseudogene where it shares 96% sequence similarity. And you can see it's the top 5% of all genes, pseudogene apparent genes. As a consequence, and this is work by Toffoli and Christos Prakakis, where you can show that then, as a consequence, when you do whole genome sequencing, you have a really low mapping quality over GBA on the right and GBA pseudogene 1 on the left. And we thought then, wouldn't this be the same for RNA sequencing? So we did the same kind of analysis. And of course, we see that of all the reads mapping to GBA, only 41% of those actually uniquely map to GBA, meaning that they only map to one location, whereas 60% of them map to multiple locations across the genome. And we look at those 60% of reads where they map, it's all to the pseudogene. Of course, we thought that this must have consequences for our understanding of these two genes. So to actually approach this, and I think this was really a timely question, is that the introduction of long-read RNA sequencing, where instead of just sequencing short reads, which cover at most individual splice junctions, you now can sequence complete transcripts in one go. So there's no inference being made here, and you can actually then first accurately assemble the transcripts, but you can also quantify them. So we then developed an approach. So this is a targeted long-read RNA sequencing. We've done this across 12 different brain regions. So you can see each brain region is a mix then of multiple samples or individuals. And this is all now in control samples just to understand the normal function, normal structure of these type of genes. We also then validate some of these in iPSC-derived cortical neurons as resides in microglia. And this is based on hybridization capture. So, of course, there's a lot of stuff that came out of this work, but I think the most important, or the one I want to emphasize today, is this, what's in green here. So if you look at the total expression then across each region for GBA on the top and GBA pseudogene on the bottom, you can see that what's in green, these are all transcripts that are predicted to be coding, but they have a novel open frame, so not in annotation. And when you look at GBA, you can see that the encoded nucleus that represent about 36% of total expression from GBA is actually novel protein coding. And perhaps more surprisingly is that we find about 5 to 10% of all expression at the pseudogene is also predicted to be coding. When you again look at the GBA, you can see that a lot of these transcripts are, on the top you have the canonical or the main select. You can see that these are shorter isoforms at the end terminus. Whereas for the pseudogene, it's just additional splicing, and they just have an open reading frame. So they're quite similar to what's in annotation. Yes, they hadn't been predicted as coding. We thought, okay, would this have a functional consequence? So what we've done here, you can see that this is a structure of our GBA with a single peptide, and then you have the glycohydrogen domain, which is important for the GCase function. You can see at the bottom here, the blue is the full-length GBA, of course. And you can see that for most of the GBA sequences, we would predict that this would have consequences for them, this to work as an enzyme within the lysosome, or even being translocated to the lysosome. But interestingly, we find that actually the pseudogene has most of this domain and part of this signal peptide intact. When we then use AlphaFold, we can see that it's actually structurally, while they're missing certain domains, they seem to fold very similar. And even here, when you look at the pseudogene with GBA, it seems to have a somewhat similar structure, actually. So what we first did is then we then express this in cells where GBA is knocked out, and we can find GBA and GBA pseudogene is knocked out, and we can see translation by measuring this with a flag tag of all of these transcripts, including the pseudogene. We also then used mass spec data, so this is public mass spec data for dorsolateral frontal cortex. And we can see unique peptides to the pseudogene here that's present in the mass spec data that we don't find actually mapping to GBA itself. So we now found this in multiple data sets. We actually have in vivo then evidence of translation of the pseudogene. And this is obviously still ongoing work, but we then look at where do these transcripts go, and we find that for most of these GBA transcripts or isoforms, we see that they actually don't seem to co-localize with lysosomes, as measured here with cathepsin D. But actually for the pseudogene, we do find that some of these transcripts actually seem to overlap with lysosomes, so about 10 to 15% of the protein potentially. And just briefly, when we then use 5' single nuclear RNA sequencing data on the top here, and we use then, again, as I said, the cortical neurons, astrocytes, and microglia on the bottom, we can see that there's some cell type selectivity to this expression, and we find that most of it seems to be in excitatory neurons and in astrocytes, and we see very little of it in microglia, and this is the same for actually both GBA and GBA pseudogene. So obviously this adds another layer of complexity of understanding these genes and loci implicated in disease, even for a gene as GBA that we've been studying for many decades now. But this paper came out about a year and a half ago, and I think, again, this was presented yesterday by Christine Klein, and it's a GP2-led GWAS of African admixed populations where they've done the case control. And what was highlighted in this is a very clear and very clean signal at GBA1. And this is a variant that has not been implicated in disease in any other populations before. And they predict that this actually would act as an expression quantitative trait loci, so EQTL. So I was then involved in this study, which is led by Cornelis Blauendrath and Pilar Alvarez-Jerez at the NIH, or was at the NIH. We then see that when you analyze based on the risk allele, which is D here, so the homozygous heterozygous, you can see that as a consequence, you have a high RNA expression here, which is quite, I guess, counterintuitive, but you see a lower translation of it, so you see less protein. So we figured this must be because of on a transcript level that we see this effect. So what we did is, and I hope you can appreciate, you have the wild type, the heterozygous homozygous. You can see that there's this retention of an intron here, partially retention. And when you look at the long read RNA sequence data, you can see this, some of these transcripts that are starting at the very end, which has partial intron retention. And when this is quantified, you have the risk allele here, homozygous, heterozygous, and a wild type. You can see that this is retaining in almost 50%. Same with this intron, but when you see the exons pre and post this intron, you don't really see an effect. So it seems to just be an intron retention effect. And important, when we then find that this risk allele then introduces disrupt a branch point here in this intron, and as a consequence, you have this intron retention event. And again, when you then quantify the GCase activity, you can see that you have a reduced activity as a consequence of this variant, which is quite similar to the other known PD risk variants in GBA. So here I then kind of showed you how you can actually use this long read RNA sequence to actually understand the consequences or get a mechanistic insight to a risk variant that seemingly was non-coding. And lastly, I just want to touch briefly on how we can actually apply this to understand, well, to improve our therapeutic designs. So I mentioned synuclein. So synuclein is the main component of these Lewy bodies, the pathology. And this can happen due to qualitative changes. So this is missense mutations in GBA, as you can see here. But it can also be due to quantitative changes where you have multiplications, duplication, triplications on the loci. Interestingly is that when you overexpress, or the more synuclein you have, the higher aggregation kinetics you see. But also, when you have different combinations, you add other isoforms, you can actually see that other isoforms can drive the canonical to aggregate faster. So here, a combination of 112. Despite this, we don't really know much about synuclein's transcription in neurons. So I'm going to skip this. It's the same protocol, but here we used midbrain dopaminergic neurons. And again, we find that a lot of variability here actually happens at the 5' and 3' end, and I exemplify that here, where you can see that there's a lot of these shorter 3' UTRs. So you thought, can we actually use this then to improve an ASO design? So again, I here plot all the UTRs, and we designed 9 ASOs here across these UTRs. So this refers to the positions. And you can see that based on this, this UTR here then corresponds to about 25% of expression, and then lower and lower. So based on this, you can see that if you use ASO1, which is here, you can see that you can knock down up to 90% of synuclein, or 80 to 90%, and then this goes down depending on where in the UTR you actually target. So we went ahead with ASO1, and we did this in neurons from patients with the A53T mutations and triplications. And again, you can see that the effect on the protein level is higher than on ASO1, and then goes lower the further out that the UTR you go. And here it's just done in SHSY cells, so you don't see the same effect, because here the UTRs really look different compared to neurons. And as a consequence, we treated this and then measured things like here, aggregation of synuclein, and you can see that in triplication carriers with ASO, we can reduce this, do the same over here, and in A53T mutations. Similarly, we can rescue cell death by treating with this ASO1. Then you can see here on this side. And lastly, we then look at the mitochondrial network, which is disrupted, and we can see we can actually improve this network again in triplication carriers in A53T mutations. So just to very briefly summarize, and I think the most important point here is probably that I think that when we start looking at these gene flows, we should probably go away from a gene-centric approach and start looking at a transcript-centric approach. Because this can then, as I showed you, we can see that a large proportion of GBA seems to be novel coding, and it does not go to lysosome, and I mean, that's about a third of actual transcription there. We can then use this to understand non-coding risk variants, or supposedly non-coding risk variants that we've done in GBA, but also we can actually use this to improve ASO design for these low cells. I think that's my time, so with that, I want to thank a lot of all the collaborators and everyone who supported this work, particularly ASAP, that's been a main funder of some of this work. Thank you.
So thank you so much also for sticking to time. Are there any questions in the audience? Maybe I'll start to warm up. I'll start with GBA, with the pseudogene.
Yeah.
Emil, are you able to speculate on, do you think that this is a functional protein? And if it is a functional protein, could you take a guess of what its function might be?
Yeah, functional in the sense that, yes, I do think it's translated. The function itself, I'm not sure. I think, to me, the most exciting aspect of that is that maybe we can actually repurpose the pseudogene for the loss of GBA itself, similar to what we've done with SMN2 and SMN1. And I think that's probably, to me, the most exciting. I mean, what it does right now, it's, yeah, it seems to be a lot of it by Golgi. So I don't know if it has a function by the Golgi, but that's kind of what we see.
Are there any functional studies with the purified GBA1 pseudogene that might support that?
No, we're trying to develop an antibody now so we can actually study this endogenously. That's, I think, step one, to see if we can actually measure it properly.
Very interesting. Yeah. Okay. Any other questions?
I might have gone over that a bit fast.
Okay.
Okay, thank you very much indeed, Neil. That's a really excellent presentation. And we're going to move on now to my co-chair, Dr. Esther Samler, who is a clinical academic, a clinical neurologist and a basic scientist working in Dundee. And Esther has done an enormous amount of groundbreaking work on LRRK2, its function and its pathology. So we very much look forward to impact of LRRK2 gene mutations on endolysosomal function. Esther.
Okay, thank you so much for inviting me. I'm very excited to be here. It's my first time in Liverpool, and it's also my first time at a British Neuroscience meeting, so thank you very much. So we are now moving from transcript to protein, and from GBA and alpha-synuclein to LRRK2. So I have no disclosures. Emil already gave an introduction to Parkinson's disease. I just want to add that Parkinson's disease is really common. There are now over 10 million people affected by the condition. We still don't have a cure. In the previous section, we heard that there is some hope and that we also have good symptomatic treatments, but as of now, we really don't have anything to offer in terms of disease modification. Importantly, it seems like we are in the Stone Ages compared to other branches of medicine. There's really no test for diagnosis, like now I'm in clinic and I have my patients finger tapping and so on. We don't have really good measures for measuring progression in patients. And we really only have a rudimental understanding of what causes Parkinson's disease. I will skip over the genetics of Parkinson's disease because Emil already talked about this. LRK2 is probably, well, I think like here we have to argue. I think like, Emil said GBA and possibly alpha-synuclein. I would clearly favor LRK2 as the most important Parkinson's disease gene, but I think like, we are all up there in one area. But LRK2 was discovered as a disease, so that gene mutations cause Parkinson's disease, was discovered in 2004, so just over 20 years ago. It was really exciting because it is a kinase, and it pretty much became clear that activating mutations in LRK2 cause Parkinson's disease. Now we know that about 2 to 4% of people living with Parkinson's disease carry a LRK2 variant, and we have developed assays to measure LRK2 activity. So in Dundee, Dario Alessi and colleagues identified that RAP proteins, a subgroup of RAP proteins, are the endogenous substrates of LRK2, and we can harness this to measure the activity of any variant in LRK2. We can do that in overexpression models, and we can also do that in patient cells. We know that LRK2 is, funnily enough, not highly expressed in the brain, but highly expressed in immune cells and also in lung and in kidney. And that makes you wonder what the endogenous function is of LRK2. And excitingly, because it is a kinase, because we think that, that kinase activation is the mode of operandi by which LRK2 causes Parkinson's disease, so it's a highly pursued target for disease modification, and there are several companies already in clinical phase trials with LRK2 kinase inhibitors, type 1 inhibitors, then an antisense oligonucleotide, similar to the strategy that Emil just talked about, and then also PROTAC degraders of LRK2. And so I mentioned that we can measure LRK2. So we can do that by looking at the phosphorylation of LRK2, of its endogenous substrate RAP10. We published a paper a couple of years ago together with Dario Alessi, where we looked at 100 variants that had been reported to occur in Parkinson patients. We expressed them in HEC293 cells in an overexpression system, did multiple replicates, and then identified that out of these 100 variants that we tested, 23 were actually activating. And this is already much more than the few variants or clear-cut mutations that had been linked to Parkinson's disease by traditional genetics. So, and we defined that LRK2 kinase activity as activating when the activity of a particular variant was above 1.5 compared to the wild type. This was rather arbitrary, but we took this because that's about the activation of the modestly kinase activating mutation LRK2 G2019S, which is the most activating mutation. You may have heard from Christine, or you may have come across this paper that was just published by her group and Katja Lohmann as the corresponding author, where they really, in painstaking work, went through the literature and identified all the different variants in LRK2 that have been identified in Parkinson patients, controls, and other diseases. And so what you can see is that most of the LRK2 variants that have been identified in patients, in Parkinson patients, are missense variants. G2019S is the most prevalent variant identified, but most of the variants, so there are like 211 missense variants, 13 have been classified as clearly pathogenic, 12 as likely pathogenic, but 186 as variants of unknown clinical significance. And everybody working in the clinic, I think, that's really the term that is geneticists or Parkinson specialists. I don't know, that makes you climb up a tree. So we have actually had this data from Christine and Katja for some time, and we have really like expanded our work on these variants. And I think this is a slide from a couple of weeks ago. I think we now have covered, and we do have functional data on all of these 211 variants in LRK2 that were described in Christine and Katja's paper. And I just want to somehow show you some of this data. So here is what you will see is what I've done is, so this is transient overexpression in HEC293 cells. That sounds very simple, but it's really tried and tested. We have at least 3 biological replicates for each variant. And what you see is here, so that so what you can see is that here is the effect on RAB10 phosphorylation, so LRK2 kinase activity, and I've ranked them from highest activity to lowest activity, so you have a whole spectrum, and we have now over 66 activating LRK2 variants, then quite a lot of over 200 and almost like 50 variants that don't really change the activity of LRK2 much, and then really interesting also missense variants that significantly decrease LRK2 function, and I think a lot will be in the structure of this. And this is an overview of all the LRK2 activating variants that we've identified so far. What is really interesting is, you can see the domain structures of the LRK2 protein. It's a really large protein, 2,527 amino acids. It has a catalytic core with the GTPA domain and the kinase domain. But actually, the activating mutations or variants in LRK2, they span the entire protein, N-terminal to C-terminal, and everything in between. And what is in green is actually that myself or my team, we've traveled the world and we've collected patient samples. And I'll show you one slide of how patient-derived data on LRK2 activation looks like. So, it's relatively easy, to get samples from patients with LRK2 G2019S or even the BASC mutation R1441G, because these are really frequent. But we actually have collected over the last 2-3 years samples from patients where I'm only aware that a particular variant occurs one time. So, for example, F1700L or a few others. So everything what is in green here, we've gone through a lot of trouble to collect these samples. And here you see how these assays look like. On the left-hand side, it's the cellular assay in HEC293 cells. What you can see is we have overexpressed LRK2. So here's and this is the effect on endogenous RAB10 phosphorylation. So here's the effect on endogenous RAB10 phosphorylation of the overexpressed wild-type protein. Here is a known pathogenic mutation. That's one of the mutations that has the most activating effect. So the Y1699Z mutation, and here plus minus a specific LRK2 kinase inhibitor. And then this was a novel variant that we identified amongst different mutations or variants that Christine and Katja had sent us. And then when we went back and asked them, could you give us a little bit more information about this variant, the F1700L, we actually saw that, that this mutation mapped to an autosomal dominant family history of Parkinson's disease. And then we were able to travel to Lübeck, and we were able to get patients samples. What is really quite important here, we really need fresh material. The blood can really only be 1 or 2 hours from venesection until we need to start isolating neutrophils and monocytes, and we've published widely on this, but this is quite remarkable. So this is the cellular overexpression data, and then here on the right-hand side, you see these are members from the same family with the Y1699C mutation. This is a control, so RAB10 phosphorylation as a readout for LRK2 kinase activity in a control person who does not carry the mutation, here in a non-manifesting carrier, and here in two family members with Parkinson's disease and the mutation. And here on the other side, you see the novel F1700L variant, and again, plus minus MLI2 treatment. And we've published on this mutation. This has never been shown before. And then, Pavel Liss, who is a very talented senior researcher in Dario Alessi's lab, so he's doing quite a lot of structural analysis using AlphaFold and published structures. And I think we now understand of how these mutations, especially in the ROC core GTPase domain, activate LRK2. That is because intrinsically, the wild-type protein tries to somehow stay, keep LRK2 in the inactive closed conformation, and these mutations basically change the structure and thereby activate LRK2 activity. So we now have looked at over 350 variants of LRK2, and this is really quite a lot of work and really not a way forward to look at all the different variants in LRK2, because I mentioned it's a really large protein. There are 20 possible amino acids, so that leaves you with 50,000 variants. And I think I have a little bit better things to do with my life than continuing to do all these one by one variants. But we are working with Greg Kuttler at the MRC Genetics Unit in Edinburgh, and he has a fantastic methodology. So where he basically, what you need is you need a saturation library, and where every cDNA, so every variant in a particular gene is barcoded, so that you can actually go back and see what transcript a particular cDNA is used for. Then you need a cell line with a landing pad where only one particular transcript is incorporated, and then you need a functional assay, and we're using for LRK2, we are using the phosphoRAP10 as a flow assay, and basically so we have we then, sorry, I should show it here. So we have the generation of the barcoded mutagenesis library. Then we integrate this into a HEC293 landing pad cell with knockout of LRK2. Then we deploy our functional assay, and we collect the cells into bins with increased wild type and reduced kinase activity. And then, because the cells are barcoded, we can go back and ask which of the variants are activating or reducing in activity. And for LRK2, we are, as I said, we are deploying our phosphoRAP10 assay, and we are actually doing the same with GBA1, and that's probably because it's also much, much, much smaller, the furthest advanced of this project. And together with Miratul, we are also doing Parkin. So I've told you a lot about LRK2 activating variants, and what about the physiological function of LRK2, and what about loss of function variants in LRK2? So why is this important? So it is important because there are disease-modifying treatments that target LRK2 in clinical practice, and they target either the degradation of LRK2 or LRK2 kinase activity. And in mouse models, preclinical models of LRK2 with either knockout or with treatment with high doses and prolonged time with LRK2 kinase inhibitors, there was a concern over the years that maybe knockout or severe inhibition of LRK2 could cause problems in the lung, in the kidney. So there was a histopathological phenotype that caused this concern. So there was then another study that looked at really large genetic numbers of LRK2. They really didn't find many loss of or any clinical details. I think they found 6 homozygous loss of function mutation carriers, but there was no available clinical details other than it was compatible with life. But actually, heterozygous loss of function variants in LRK2 were reported to be quite frequent, and it was said that it was not, there was no discernible effect on survival or health. And over the last few years, one of my main collaborators is Alexander Zimbrich, who is also one of the first authors to have discovered LRK2. And he and a wonderful geneticist at the University of Istanbul, Tuba Kalachi, so basically they came together on Matchmaker, almost like a dating platform for geneticists. And we found a consanguineous family in Turkey where 2 brothers suffered from carry nonsense, homozygous nonsense mutations in LRK2. And at that time, when we came across the mutations, we didn't really know the phenotype, but we actually got in touch with her. And then we heard that these brothers suffer from young onset lung fibrosis. And that was really interesting for us. And what we did is Alexander and I, we worked with our Turkish colleagues, and we traveled there last year, and we were able to bring the family, so the 2 parents, and they have 4 children, including the 2 with lung fibrosis. We were able to bring them to Istanbul, and we collected different biomatrices, including peripheral blood neutrophils that we isolated there on site. And what you see here at the top, you can see that So if you look in the middle, there's LRK2. You can see that there were actually 3 individuals who did not have any measurable LRK2 in neutrophils that expressed really high levels of LRK2. And on the left-hand side, you see RAP10 phosphorylation. So again, like these 3 individuals did not have any endogenous RAP10 phosphorylation. This was measured by one of my postdocs, Sarah Gomez, by targeted mass spectrometry for LRK2 and also phospho-RAP10. And then underneath, you see the Sanger sequencing. So actually, like what we did not know, what only became clear here, was that one other unaffected brother also carries homozygous nonsense mutations in this gene. He does not want to undergo further investigations, but we would clearly say that he's probably at risk of lung fibrosis. I'm not going to go into much more detail. Well, I have one more slide here, but we actually identified a few other families with either compound heterozygous variants in LRK2 and lung fibrosis, but obviously it is not clear whether that's monoallelic or biallelic, but we found at least 2 other cases with homozygous nonsense mutations in people with lung fibrosis, but we don't have any biological samples from these. Unfortunately, this slide doesn't come, something is missing here, but the gist is, so we can also use urine BMPs as a readout for downstream LRK2 function. What we saw is that urine BMPs in these heterozygous mutation carriers, what you would see here. So if we just look at one species of BMPs, you can see these are unrelated, either Parkinson patients or unrelated healthy controls, where you see BMPs, and then you see here the 3 family members with heterozygous loss of function of LRK2, and then the homozygous loss of function mutation carriers. So you really see a dose-dependent effect on LRK2 on urine BMPs. So, and urine BMPs are these negatively charged phospholipids that are quite important as a biomarker for LRK2. I only have like a couple of more slides. So what about LRK2? Pathway activation with other monogenetic forms of Parkinson's disease. So we previously identified, also together with Alexander Zimbrich, that VPS35, D600TN, which is a retromer component and therefore also intrinsically linked to the endolysosomal pathway, that this mutation also activates the LRK2 pathway. And I told you about BMPs and the loss of function mutations, that urine BMPs in these very rare patients are reduced. But in LRK2 mutations, these urine BMPs are elevated, and not just in LRK2, but also in VPS35. But what's next for unraveling endolysosomal dysfunction in Parkinson's disease? So we developed, maybe I should start with one problem with looking at endogenous lysosomal or endolysosomal function in patients is really that lysosomes make up only like 1 to 2% of a cell's content. And looking at lysosomes endogenously is really difficult. So we developed a methodology for immunoenriching intact lysosomes from peripheral blood. We worked with Monta Aburamaei at Stanford University, who is part of our ASAP team. And rather than going for Parkinson's disease in the first instance, we actually tried to show proof of principle in children with the lysosomal storage disorder, CLN3 button disease. And what we saw is, we saw a massive lysosomal enrichment of glycerophospho-D esters. And if you just look here on the left at the GPG, so what you see here in this lysosomal fraction with our methodology in healthy controls compared to the patients, there was a massive enrichment of these metabolites. But when you looked at the whole cell lysate level, you were not able to somehow see this difference. So this is now a way of really closing or zooming in on the lysosome. But a problem with this lysoTARC or TARC-less lyso IP approach was that we used a commercial antibody from Upcom. And since we published our paper, Upcom has increased the price and basically removed the freedom to operate for us to use to really like make the lyso-IP method widely available. So we've really frantically worked on generating an in-house antibody. So again, like, that was in Dario Alessi's lab, Pavel Liss in Mindaugas Viscontas have done a lot of work. And then me and my group and my clinical collaborators, we've validated this. And this was really with lightning speed. So here, I thought I need to somehow validate the effectiveness of this antibody also in CLN3 patients, which are very rare. So here I'm working with Peter Hasselt, he's in Utrecht. So Sarah Gomez, who's one of my postdocs, so we were in Utrecht at the end of January. We then brought the samples, we collected samples, performed lyso-IPs in Utrecht, then traveled home with everything in dry ice in our bags. And then Dario Alessi, he then the next day, we processed the samples, and the next day he then flew to Stanford, and then here the samples soundly arrived on dry ice in Monte Aburama's lab, and this is Eshan, a wonderful postdoc who's been working with us. And this is just to somehow show that, so we now have really like wonderful data to show that our C1, sorry, I don't know why there's an exclamation mark, our C1 antibody is as efficient as the upcome antibody in enriching lysosomes. And what's next? We are now applying this to Parkinson's disease. Maybe some of you have heard already of the LRK2 LIGHT, the LIGHT initiative, the LRK2 Investigative Therapeutic Exchange Program. So this exists out of 3 pillars, and I'm not going to go into detail. There is information on the web, but basically, pillar 3 is the development of LRK2 pathway relevant clinical biomarkers, and we will use the lysoTAG approach to look at LRK2 driven lysosomal biomarker development in really like quite a large proportion of monogenic Parkinson's disease subjects that we are recruiting from across the world. And so I'm very, very excited to see what is to come. But now this is my most important slide. I really need to somehow thank all of our wonderful collaborators. Sorry, that's in the next one. I want to somehow thank all of our wonderful collaborators from healthy volunteers, from patients, from all the clinicians who work with us, and then all the different members of the different teams who make this possible. And I'll just leave my summary slide up there to then take questions.
Thank you very much, Esther, for a wonderful presentation. I've got a couple of questions, but I should ask the audience if there are any questions. Please raise your hands.
Thanks very much. Hopefully you're getting your steps monitored while you're doing this. Esther, thanks for a lovely talk and wonderful data. I was wondering if you could speculate on whether you think one of the RABs is the important one for disease pathogenesis. So you've got a host of different RABs which are phosphorylated, a couple which are genetically linked to PD which aren't phosphorylated. And I was wondering whether you see this as a single pathway downstream of LRK2, or whether there's a pleiotropic effect or something along those lines.
So that's a very good question, and the question is like that we don't know that as yet. I think what might be quite, so at the moment I'm using RAB10 because we have the tools to study RAB10, because we have this wonderful phosphor-specific antibody against RAB, the threonine 73. But I could imagine, but I can't prove that as yet, that maybe like phospho-RAB12 may be very, very important, because that may be very relevant in the brain. And at the moment, we have phosphor-specific antibodies, but that are not really particularly well. But I think, by developing a targeted mass spectrometry assay, what we are in the process of doing, that will help us a lot. And I think, for me, obviously, I'm a clinician, and I work with the samples that are available for me. So I need fresh material. So the periphery is quite important. I've done some work in postmortem brain samples, but to look at post-translation modifications is really difficult. So I don't know as yet a way around how to look at phospho-RAB12 in the brain in humans. Thanks for the question.
Hi, thank you. Yeah, that was a really beautiful talk, and my question's quite related actually, because a lot of researchers now kind of pinpointing RAB dynamics as having a role in Parkinson's pathology. But do you think this will also be for other neurodegenerative disorders, and RAB changes being involved there?
I feel really bad because I have to answer to every question. I do not know. I do think, maybe other neurodegenerative disorders, I do not know, but I think that there's probably shared pathway pathology or dysfunction, at least in other genes that map into the endolysosomal system. So definitely VPS35. I think the question for RAB32 is still out. We know that there's an interaction, between GBA1 in the lysosome and LRK2. So I think I would start with those close cousins of LRK2 first, and then maybe branch out a bit further. But I think what would be very interesting is to look at other MSA or PSP, so PSP, where Hugh Morris has done work to somehow show that a particular SNP drives the progression. So I think that would be very, very interesting.
Thank you very much. I think we had better move on. I'll save my questions for later, Esther.
So, and I think, so I now, it's my pleasure to introduce my co-chair, Tony, and he will talk to us about therapeutic approaches targeting the lysosomal enzyme glucocerebrosidase. And probably all of you know of his, really incredible efforts to, to bring clinical trials and testing of for GBA1 to patients. So I'm very excited, and I very much look forward to our next talk.
Thank you very much, Esther, and thank you again to Parkinson UK for their kind invitation to participate in this symposium. So I'm going to talk about therapeutic approaches targeting specifically glucocerebrosidase. And just as a reminder, the glucocerebrosidase gene I'm going to shorten to GBA1, and the enzyme to GK. So that will help inform you about my slides. Now, the pathogenesis of Parkinson's disease is undoubtedly multifactorial, and there are a number of important pathways involved in what ultimately determines the pathology of Parkinson's disease. And these include mitochondrial dysfunction, inflammation, protein aggregation, etc. But importantly, lysosomal dysfunction, which you see here, and endolysosomal dysfunction. So these are two of the several pathways that are involved in Parkinson's disease. And of course, these don't necessarily have to be exclusive, but may be present in any one individual in more than one form. Now, in terms of glucocerebrosidase, just to remind you of the pathway that we're talking about here, this is a ceramide pathway, a hugely important pathway in biological function in cells. And I'm going to be talking here about glucocerebrosidase, the big blue arrow pointing to that. But you'll see that this pathway is decorated with a number of red stars. And each one of these is a neurological disorder. So abnormalities of this ceramide pathway are really important in neurological disease. Many of these neurometabolic disorders present in early childhood, and indeed, glucocerebrosidase deficiency can as well, and I'll come to that in a second. So this is just a reminder of the pathway and its importance. The GBA1 gene, which encodes this enzyme, when present in homozygous or biallelic form, causes Gaucher disease, which is an autosomal recessive disorder, and Miller's already pointed this out. Now, Gaucher disease is a lysosomal storage disorder, as I say. It's autosomal recessive, so biallelic mutations. And it may present in infancy or late adulthood. Those cases that present with neurological dysfunction tend to present very early. And in terms of what is affected in patients with Gaucher disease is the hepatosplenomegaly, but a multitude of other dysfunctions, including bone and hematological. Fortunately, treatment is available for patients with Gaucher disease in the form of enzyme replacement therapy, which improves non-CNS organ dysfunction. And it was an interesting observation that people with Gaucher disease, but perhaps even more interestingly, their apparently asymptomatic first-degree relatives were more prone to get Parkinson's disease than one might expect by chance. And this led to the observation that GBA1 is associated with Parkinson's disease. Now, variants in the GBA1 gene are present in less than 1% of the general population, but they are, when present in any individual, increase the risk for Parkinson's disease about 20 to 30-fold. They are the most common genetic abnormality in Parkinson's disease and present in 10 to 15% of all people with Parkinson's disease. And if you look at the Ashkenazi community, that can be up to 31% of Ashkenazi people with Parkinson's disease carry a variant. But interestingly, not everybody who carries a variant gets Parkinson's disease. Indeed, by age 80, only about 30% of people who carry a GBA1 variant develop Parkinson's disease. It's not a dose effect, because the risk between those who are homozygous with Gaucher disease and those who are heterozygous is approximately the same. So it's not a dose gene effect. Now, just to remind you again about Parkinson's disease pathology, an important morphological abnormality is the presence of the Lewy body, which is aggregated alpha-synuclein, as well as many other proteins, but importantly, alpha-synuclein. So it is thought to be the increased level of alpha-synuclein and its aggregation and accumulation and formation of Lewy bodies, which helps precipitate the different biochemical abnormalities that result in neuronal degeneration and neuronal loss. It's accepted that alpha-synuclein accumulation is not the only marker of Parkinson's disease pathogenesis, but it is an important one. And you remember from earlier this morning that it is a target for therapeutics in its own right in terms of alpha-synuclein antibodies to clear alpha-synuclein from the brain. There is an interesting relationship between glucocerebrosidase, the enzyme GCase, and alpha-synuclein. Interestingly, it was shown that in the brains of people who died with Parkinson's disease with GBA1 mutations, there was a reduction in GCase activity. This is perhaps not surprising given that they carried the mutation in the gene. But what was more surprising was that people who died with Parkinson's disease also had GCase deficiency, even if they did not carry a GBA mutation. The deficiency was greater in those that did, but still present in those that didn't. And this led to what we now understand to be the reciprocal association between GCase activity, the enzyme product of GBA1, and alpha-synuclein. Anything that reduces GCase activity results in an increase in alpha-synuclein. And anything that results in an increase in alpha-synuclein results in a decrease in GCase. So you have this self-amplifying cycle present in the Parkinson's disease brain, where those with GBA mutations have reduced GCase activity and get increased alpha-synuclein. In those without GBA mutations, you have an increase in alpha-synuclein, which results in a decrease in GCase activity. So this is a self-amplifying process which has an impact on many other biochemical pathways in the brain. But the principle behind the therapies targeting the GBA pathway is that the assumption, if you increase GCase activity, you will reduce alpha-synuclein and perhaps reduce toxicity. So this is the basis of many of the therapeutics that I'm now going to discuss. So there are several different strategies to approach people with GBA1 mutations. But importantly, we should remember that Parkinson's disease patients, even without GBA1 mutations, have reduced GCase. So anything that we develop that will increase the enzyme's activity may be beneficial to those not only with GBA1 mutations, but even those without. So an obvious target is the gene itself. So gene therapy, and I'll discuss that briefly. Chaperone therapy, which I'll discuss in more detail. HDAC inhibitors have not yet been developed for this. Enzyme replacement therapy, such as is used in Gaucher disease, does not cross the blood-brain barrier and so is not going to be helpful in Parkinson's disease. However, people are looking at forms of enzyme replacement therapy in carriers that may be able to cross the blood-brain barrier and so could be helpful. And finally, there's substrate reduction therapy, the target being a reduction in the buildup of substrate that is secondary to the enzyme deficiency on the basis that this buildup of substrate is pathogenic. So, there are many different trials going on, which is really encouraging. Only one has been completed so far and published, which is the Ambroxol study I mentioned briefly. But others are on the way and others should report out. I know one which is about to publish this year. Others will report this year and next. So this is a very active area and runs from phase 1 through to phase 3, which I'll show you. So let's start with the GBA gene therapies. There are basically 3 different principles here. The first is gene replacement, to take the normal GBA1 gene on AAV and inject it into the brain. We'll actually inject it into the CSF. And the AAV with the normal GBA1 gene and then transfects into the patient's brain. So this form of therapy developed by a company called Prevail, who have started the so-called PROPEL study. This is a phase 1/2A study, which means that the most important readout is safety and tolerability of this. It's a one-off injection into the fluid that surrounds the base of the brain. This trial is ongoing and recruiting late-stage Parkinson patients. Next is gene replacement therapy, so the same principle, but a different route. And this uses an AAV capsid that is capable of penetrating the blood-brain barrier, so it's injected systemically. This is about to start a phase 1/2A trial. And the third, that has not yet entered clinical trial, is the specific mutation correction. So there is a technique now, rather like CRISPR, for correcting in vivo a specific genetic mutation. And this is currently being developed for those with the L444P mutation. There is some further basic science to be done, but that may enter phase 1 trial in a couple of years. So different strategies for targeting the GBA1 gene. Let's move to the chaperone therapy. This is a more active area, probably because it's easier. There are different types of chaperones. You're familiar with molecular chaperones. They may be simple binders, but they may be inhibitory or activating. An inhibitory chaperone, ambroxol, is the one that has been most widely studied for GBA1 Parkinson's disease, and I'll talk about it in a little while. There are neutral binding antibodies, that is to say, those that just bind and chaperone from the endoplasmic reticulum into the lysosome. And there are GCase activators, and there's a trial ongoing for that too. Let me talk to you about ambroxol. So ambroxol has been recognized for many centuries to be biologically active, coming from this plant here, Adatoda vesica. And in Indian medicine, this has been used as a drug or a plant to help shortness of breath or asthma, and carries the rather nice German translation of Indisches Lungenkraut. Now, ambroxol, interestingly, was identified on a multiple screen as an activator of GCase activity. So this screen was undertaken by a group in Toronto looking for potential treatments for Gaucher disease. So what compounds among its mass screening could increase GCase activity? And this compound, ambroxol, that came from the plant, which had been present for many years over the counter as a drug for coughs, a cough medicine, was found to be the most potent activator of GCase activity. So this is obviously a candidate for potential use in those with GBA deficiency in Parkinson's disease. So over the last few years, we and others have been looking at the action of ambroxol in a number of different models of people with GBA1 mutations in their fibroblasts, in Drosophila models with GBA mutations, in iPSC neurons derived from GBA1 Parkinson patients, in mice with GBA1 knock-in mutations, and in non-human primates, just to look at the penetrance. The summary of all of this work over several years is that ambroxol is capable of increasing GCase activity and protein content in all of these models, and in those models, such as the fly and mouse, are capable of reversing motor deficits and pathology of Parkinson's disease, including reducing alpha-synuclein levels. The mouse and NHP studies confirmed that oral ambroxol is able to cross the blood-brain barrier and activate its target within the brain. This basic science led to the first human clinical trial in Parkinson's disease of ambroxol, which is a small study, again, a 2B study, but looking at safety and tolerability in the Parkinson population. It's non-blinded. And the drug at 1,260 milligrams daily was well tolerated and safe. A number of secondary readouts, but remember this is a non-blinded study. Importantly, it did not interfere with Parkinson medication. People on the drug had a significant improvement in MDS-UPDRS, but remember, this is a non-blinded, non-placebo study. And importantly, in terms of biomarkers, there's an increase in GCase activity in the CSF and an increase in alpha-synuclein, which you would hope for as well if alpha-synuclein was cleared from the brain. So this has led to the phase 3 study, which started last month here in the UK, and it seeks to investigate the action of ambroxol over 2 years in people with GBA1 mutations and those without GBA1 mutations but with Parkinson's disease. This trial will last for 2 years in terms of recruitment and 2 years follow-up. So this is the first phase 3 study of a genetically targeted drug for Parkinson's disease. And it's important to thank all those people, those organizations that have supported this study. I mentioned that there are several other studies in this pathway ongoing, and these are the ones using ambroxol, many of which will read out over the next 48 months. So we will begin to get an idea of whether ambroxol is useful for Parkinson's disease, but we'll probably have to await the phase 3 result for a definitive answer. There are GCase activators. So this is a drug that is from Bial. This is the results of a phase 1b study which showed it is reasonably well tolerated. And the drug has now just completed a phase 2 study looking at people with Parkinson's disease who have GBA mutations. And it is a relatively long study in terms of total exposure of 78 weeks. But the first readout will be much before that. This trial has now completed recruitment. So again, more activity in the GBA space. So let me finally turn to enzyme replacement therapy, to substrate reduction therapy. This, remember, is based on the premise that the accumulation of the substrates of the GCase enzyme are bad. The study using this drug venglustat, which is a glucosyl ceramide synthase inhibitor, so it reduces the formation of the substrates, was tried in Parkinson's disease and was published, but unfortunately did not work over the period of drug exposure. In fact, patients did rather worse on this drug. And the reason for that probably lies in the fact that this drug interferes with dopamine release. So the patients who are on dopamine therapy, their drugs were not effective, and you'll see that resulting in this rapid worsening of their symptoms. So, I've taken you on a tour of therapies of the GBA1 pathway. This is a very active area. People are now focusing on the chaperone space in particular, but also on the gene therapy space. And the potential for this pathway is that it is relevant not just to those who carry GBA1 mutations, who, remember, are a significant proportion of those with Parkinson's disease, 10 to 15%, but also those potentially without GBA mutations who nevertheless have reduced GCase activity. Thank you.
Thank you so much for this wonderful and really informative talk. Are there any questions in the audience for Tony? Oh yeah, there's a question. Oh, more questions. Wonderful.
Thanks for the presentation, Tony. Very interesting. GBA mutation carriers have a greater preponderance to develop cognitive decline. What do you think is the mechanisms behind that?
Thank you, David. So I'll speculate, because we don't know for sure, but I speculate that it is the increased accumulation of alpha-synuclein. So that self-amplifying pathway is already triggered by reduced GCase activity. And you would expect that the lower the GCase activity that results from the mutation leads to a greater accumulation of alpha-synuclein. And there are post-mortem data to correlate with alpha-synuclein Lewy body accumulation and reduced GCase activity. Also, those people who carry the severest GBA mutation, who have the lowest GCase activity, tend to get the greatest cognitive impairment earliest. So I think the data suggest it's associated with greater alpha-synuclein accumulation.
Tony, hi, thank you. That was a really super talk. Looking forward to seeing those phase 3 studies go through with ambroxol. But my question is really about the venglustat study, the reduction that was noticed there in the patients performance. I don't, as a preclinical scientist, never going near a patient in my life, I just wonder how venglustat got through to clinical trials if it was known to reduce dopamine release. So could you please comment on that?
Yes, a very good question. Well, the speculation that it reduces dopamine release is speculation. So it was not known to have any direct effect on dopamine, nor on dopamine medication before the clinical trials began. It had already been developed for Gaucher disease, for which it is potentially quite effective and safe. So the company developed the venglustat trial for Parkinson's disease, and I think everyone was surprised to see that there was this reduction in mobility in the UPDRS, and that it developed quite early on in the study. So it's not due to increased degeneration, which you would expect later. And if you remember from the graph, so that the worsening developed by the first observation point and didn't get worse after that. So I was involved in the study. We have been looking at what happened to the patients longer term. They haven't got worse, so it's definitely not increasing neurodegeneration. And other studies are looking into whether this drug really does impair either the synthesis or the release, or alternatively directly impairs the absorption of the drug from the intestine.
Yeah, okay, there's a question here.
I'm just interested in terms of how this could be applied to other people with synucleinopathies. So we know that there are 5' and 3' SNPs for synuclein that lead to increased synuclein, granted low levels but lifelong, and then people with duplication and triplication. Do those people have modified GCase?
Very interesting question. No one's really looked at that properly. I mean, people have looked at some MSA cases. I can tell you that those with triplications do, or at least their iPSCs do, because we've looked at that. So the answer is probably yes, they do, on the basis of the reciprocal relationship. So they have probably got GCase activity reduced. I will say that in MSA, GBA mutations are not increased. In most studies, they are in some, but not very significantly. But looking at GCase activity in MSA has not really been done properly. So the short answer is that if the drug happens to be, or any of those GCase activators happens to be helpful in Parkinson's disease, they may certainly be applicable to other synucleinopathies.
Any other questions?
Thank you, very nice talk. I was just thinking about the correlation or interrelationship between alpha-synuclein and GCase. So in Parkinson's patients, certainly at the later stages, do these patients have hepatic enlarged livers and enlarged spleens as well? Because that would be a logical conclusion, I guess, from the interrelationship.
Yeah, no, they do not. And the basis for that is that the reciprocal relationship is only present in those cells or areas where one or other is active. So, for instance, people who have heterozygous GBA1 mutations tend to have systemic moderate GCase loss, down to 50%, because they've got one normal allele. That is probably enough systemically to not result in any biological increase in relevant increase in substrate. In the brain, however, say within a neuron where there's only 50% GCase activity, that is sufficient to result in alpha-synuclein accumulation. So the reason why the Parkinson patients with GBA1 mutations do not get any Gaucher-like problems outside is because you've got to have an intracellular relationship between those two.
therapy. Has anybody, and maybe I missed it, has anybody looked at, particularly from the CNS symptom side? So there's a company in Japan, JCR, who've got a drug approved called escargot, where they have used a hijack protein essentially on the blood-brain barrier to treat Hunter's disease, MPS II. So has anybody looked at that approach for addressing the CNS symptoms with an enzyme replacement therapy?
Yes, they are looking at that, but it's not yet reached clinical trial.
Okay, I can't see any other. So I'm so sorry. There was, I think, like one of the first hands raised. So that's our last question, and I'm so sorry for.
Thank you so much. That's perfectly fine. So I have a question about the gut-brain axis in Parkinson's. Do you think that if we assume that Parkinson's does start in the gut, do you think that GCase enhancement therapies could be beneficial at these very early stages for balancing out that alpha-synuclein GCase interaction? Thank you.
Well, very interesting question. It's got a few parts, and I'll try to be very brief on this. The gut-brain axis, is it relevant to Parkinson's? I think probably it is. But not to all Parkinson patients, but to a proportion. And I think there's this perception that there may be so-called body first, where alpha-synucleinopathy arises in the gut, and brain first, where it arises in the brain. And the two finally come together, but the early prodromal periods may be different. And in those with so-called body first, there may be a change in the gut microbiome, which leads to an increase in alpha-synuclein formation, probably through inflammatory change in the gut. Increased localized alpha-synuclein formation, particularly in the enteroendocrine cells in the gut, which have a monosynaptic connection to the vagus, then allows the alpha-synuclein to be transported from the gut through the vagus to the dorsal motor nucleus and to the brain. And this has been confirmed in several different animal models. So I do think that's important to a proportion of Parkinson patients. Therefore, those who carry a GBA1 mutation before they develop Parkinson's disease may be very suitable targets for therapies that increase GCase activity, or alternatively to modification of the gut microbiome that reduces the pro-inflammatory, pro-alpha-synucleinopathy that may develop in the gut. So this area is being very actively researched at the moment. But yeah, a very interesting question.
So thank you so much. I can't help but adding that there's a link between LRK2 and the gut. So all the work done by Inga Peter at Mount Sinai, so she was the one to identify variants in LRK2 in the kinase domain N2081D that are a risk factor for inflammatory bowel disease. So it's a fascinating topic. I'm so sorry. But I'll hand over to you for the introduction of the last speaker.
Thank you very much, Esther. So it's a great pleasure to invite Sarana Fanning to talk to us about lipid metabolism as a therapeutic target for Parkinson's disease. Sarana has undertaken groundbreaking work in the area of lipid metabolism and neurodegenerative disease. So this is going to be a very exciting talk. Thank you.
Okay, well, good morning. I'd like to begin by thanking Parkinson's UK for the invitation to be here and to share some of the therapeutic targets that my lab is currently working on. And so I frequently start my presentations by showing this image and asking people what difference they see between the image on the left and the image on the right. And people will often tell me that they see an image of a brain in the image on the left and no brain in the image on the right. But as it turns out, there's a brain in both images. But in the image on the right, the brain is now devoid of all lipid content. And this is because it's been put through a method known as clarity that removes all of the fat from the brain. And I think it's a particularly important image to show to convey just how lipid-rich the brain is and why, for any of us studying brain diseases, we should be considering the role of lipids. And so, to begin with, Jeffrey Kirshner earlier this morning and Tony Shapiro told us about these Lewy bodies in Parkinson's disease, these hallmarks of the disease. And for many years, we've thought about these hallmarks of disease as being accumulations of misfolded alpha-synuclein protein that you see right here. And my group and others in the field still believe this to be true, of course, but now we're thinking about the idea that it may not just be that Parkinson's disease and other synucleinopathies are simply proteinopathies, but they may be coupled also as lipidopathies. And there are several reasons for this. So the first is that relatively recently, in the past 5 years, we now know these Lewy bodies to not just be accumulations of the alpha-synuclein protein, but to also contain other components like lipid membranes, which you see here. We know that the alpha-synuclein protein binds lipids and fatty acids. And as more and more GWAS are performed, we know that there are increasing numbers of lipid and fatty acid genes implicated in risk for PD. And we also know lipids and fatty acids to be altered in PD patient samples, so things like tissue or CSF or plasma. We're not yet at the point where these have all converged on a lipid biomarker, though. And for the purpose of today's talk, I'm going to tell you about how the alpha-synuclein protein and its expression impacts the lipidome. And so when we started this work, we performed very basic experiments where we overexpressed a vector versus alpha-synuclein in primary rat cortical neurons and performed lipidomics. And really, we just wanted to see what the impact of the alpha-synuclein protein was on the lipidome. So when we look at this heat map, what we were looking for was changes from blue to yellow or yellow to blue. And what really stood out to us is here, as the neurons age, or as you increase the amount of alpha-synuclein protein, you get this change from blue to yellow in lipids designated to be triglycerides. And so in order to work out why alpha-synuclein might be impacting triglycerides, we had to look in a more detailed fashion at the lipid pathways. And what we noticed is that the triglycerides are a fundamental component of lipid droplets. And essentially their function is to store these fatty acids that come from upstream to prevent those fatty acids having toxic consequences in the cell. And so we started to ask at this point, what's happening upstream? What are these fatty acid precursors that may be dysregulated that cause the cells to require these triglycerides to store them? And when we performed fatty acid profiling, what we found was that of all of the fatty acids we profiled, monounsaturated fatty acids were specifically increased in response to the alpha-synuclein expression. And I'm showing you one of them here, oleic acid. So you get this doubling in oleic acid in response to alpha-synuclein. And so then we asked, well, is it that there's some unique interaction between oleic acid and alpha-synuclein? So we took these alpha-synuclein expressing primary rat cortical neurons and asked, does oleic acid alter their viability? And as it turns out, it does. So you can see by adding oleic acid to alpha-synuclein expressing primary neurons, you reduce their viability. And already at this early stage, we were starting to think about therapeutic targets. So we asked, well, what if we reduce the ability of the cells to make this oleic acid? Would this rescue the viability defect? And it turns out it does. So there's a gene It's called steryl-CoA desaturase, or SCD, and it's responsible for synthesizing oleic acid from stearic acid. And so if you reduce the ability to make SCD, you can rescue the viability defect in these primary neurons. And so at this point, we transition to various human cell models. And what you can see here is, in a similar experiment, even at a very low concentration of SCD inhibitor, you start to rescue this viability defect in human neurons. And then we move to Parkinson's patient-derived iPSC neurons. And where you see PD here, these are patients that have a triplication of the synuclein locus, and right next to them are the corrected neurons. And so we simply asked, can we start to reverse phenotypes associated with these PD neurons? So in this case, we take the PD neurons, we treat them with an SCD inhibitor, and what we find is that you can reverse the abnormal accumulation of the unfolded protein response that's associated with triplication of the synuclein locus. And as was mentioned earlier, one of the key problems in Parkinson's disease is this aggregation of misfolded alpha-synuclein. And so we like to model this in several ways in cells, one of which is to use what we call 3K alpha-synuclein. So this exacerbates the E46K alpha-synuclein mutation associated with familial PD. And it results in these green foci, these alpha-synuclein aggregates or inclusions that you see here in the diagram, or sorry, in the image. And what you see is that as you increase the amount of SCD inhibitor, you dose-dependently decrease alpha-synuclein aggregation. And then the final step at this point was to look in vivo in a C. elegans model of alpha-synuclein toxicity. And so these worms expressing alpha-synuclein have increased levels of degeneration of dopaminergic neurons. But if you knock down or reduce SCD, you reduce this degeneration of dopaminergic neurons. And so over many years, we used many different cellular models and patient-derived neurons, but the work ultimately culminated in a mouse in vivo study. And this study is premised again on the 3K alpha-synuclein. And this 3K mouse, developed by Dennis Selko and my colleague Silke Neuber, recapitulates a lot of the phenotypes we associate with PD. So it has motor deficits, it has neuropathology in its brain. If it ages over 12 months, you start to see Lewy body-like structures in the brain, and it has a tremor. It's also responsive to dopamine, and there are sex differences in this 3K alpha-synuclein model. And one of its motor deficits means that it can no longer climb down the pole. And so one of the questions we asked as part of this study was, if we treat this 3K mouse with an SCD inhibitor, will it be able to climb down the pole? And so what you see here is that once this mouse has corrected the level of monounsaturated fats, you can now see it climb down the pole. And we've just had a paper accepted recently that shows this SCD inhibitor also works for mice with GBA mutations and also for patient-derived neurons with GBA mutations. And so relatively recently, this has been acquired by a company. It's entered clinical trials, and so we're interested to see how that will look in the long term. But as we think about patients being diagnosed at different points in time and perhaps having different genetic mutations, it's possible that correcting their lipid imbalance in different ways would be wise. And one of the things that we noticed in doing microscopy during the SCD study was that when you overexpress alpha-synuclein in any model, so here in yeast, in the primary rat neurons, or in human neurons, you get this huge accumulation of lipid droplets. So again, signifying that in response to alpha-synuclein, the cells have to store up some fatty acid that otherwise would be toxic to the cell. And so in one way, this made a lot of sense. Biologically, in the brain, the central nervous system very tightly regulates fatty acid synthesis and metabolism. But in another way, it was something to consider for additional candidate therapeutic targets. And the reason I say that is, if I bring you back to this pathway that I showed you in the very beginning, it's possible that if you consider this brain and the orange dots being lipid droplets, and you consider this brain to be the brain of a 20-year-old who in many decades may go on to be diagnosed with PD, it's possible as this person becomes 20, 30, 40, 50, 60, these lipid droplets will accumulate over time because of the synthesis pathway has been acting over decades. And we know this accumulation of lipid droplets to be true in the brain based on my colleague's work, Ole Isaacson. And so because of this synthesis pathway, you have an accumulation of lipid droplets over time. And so it's likely that at some point, because the brain so tightly regulates this lipid and fatty acid synthesis, it may do exactly what it's programmed to do. And that is to use these fatty acid stores and break them down to generate fatty acids that can then go on to be incorporated into phospholipid membranes. Why do we worry about that? Well, because in the same way as the fatty acids generated here can go on to be incorporated into phospholipid membranes and cause problems for the disease protein interacting with those membranes, these fatty acids have the same potential. And so in the same way as we looked at SCD for the synthesis pathway, we wanted to consider a lipase in the degradation pathway as a candidate therapeutic target. So the first thing we did was to use this 3K alpha-synuclein aggregation cell model. And again, you see that applying a LIPE-specific inhibitor dose-dependently reduces alpha-synuclein aggregation. If we look at our patient triplication neurons, again, applying this LIPE inhibitor as a treatment corrects the abnormal accumulation of components of the unfolded protein response. And in the C. elegans model, you can rescue the degeneration of dopaminergic neurons by reducing the amount of LIPE activity in the worms. And so mechanistically, as we had done with the SCD inhibitor, we needed to know what fatty acids are changing and where they're changing in the cell. So we performed lipidomics and we asked quite specifically, what are the phospholipids in which 18.1 is incorporated that are reduced upon LIPE inhibition? And we find that lipid classes like phosphatidylserine or PEO are reduced in response to this LIPE inhibitor. We think this is important because these lipids play really important roles in signaling and in membrane structure and many other functions in the brain. And more specifically, what we've been able to do is not just look at things on a lipid class level, but bring it down to species level. So this includes looking at the phosphatidylserine species and noting that species like 36.2, for example, are reduced in response to the LIPE inhibitor treatment. Why does this matter? Well, we know from various studies that species like PS36.2 are increased in the brain of PD patients. And what we've been able to do is further reduce this to subspecies level to know exactly the fatty acid identities of PS36.2. And so we've now transitioned this through the 3K mouse synuclein model that I showed you earlier. And it turns out that reducing LIPE in these mice can restore or can correct motor deficits and neuropathology in these mice, but specifically for males and not for females. And we think this is really interesting because we know there are sex differences in Parkinson's disease. And so we'll try to use this as a model to work out what those differences are over time. And so this really brought us to the point of, what would you do? So we now have an inhibitor of the synthesis pathway, SCD, and we have an inhibitor of the degradative pathway, LIPE. And so if you put yourself in the shoes of a physician or a clinician, what would you do for your patient that walks into your office and you diagnose them with Parkinson's disease? Are you physician A, who decides that you want to stop this increase in oleic acid production, knowing that potentially over many decades, your patient may have accumulated lipid droplets in their brain that may get broken down because the brain decides to regulate the lipids in the CNS system? Or are you physician B that thinks your patient may have accumulated these lipid droplets over time, and therefore you should treat them with a lipase inhibitor, knowing that, of course, this pathway will still be active? So one of the things we then moved on to consider is this idea that perhaps there's a physician C, a physician who thinks that you should suboptimally dose your patient and target both pathways or co-regulate both pathways so that a patient could be treated over decades. So does this work? Well, again, we started with our 3K alpha-synuclein aggregation model. And here what you see is that when you treat with a LIPE inhibitor or an SCD inhibitor, you reduce the aggregation of synuclein. If you treat with both, this reduction in synuclein aggregation is additive. And we needed to know what the fatty acid factors were that lead to this phenotype rescue. And so when we take these cells and we fatty acid profile, we find that in fact the only two fatty acids that, if you like, are additively reduced in response to this co-regulation are two monounsaturated fatty acids, oleic acid and palmitoleic acid. And then we wanted to test this in patient neurons. So again, here we're taking the PD patient neurons that have a triplication of the synuclein locus, and this time our readout is phosphorylated alpha-synuclein. And what you see is that treating with both inhibitors at the same time gives the most benefit in terms of reducing phosphorylated alpha-synuclein. And so we think that we have two ways, either through synthesis or degradation, of targeting many of these PD phenotypes, and that perhaps a co-regulation strategy is a good approach. And all of these strategies and approaches are premised on correcting synuclein to membrane interactions. So we thought about potentially altering these membrane interactions or membrane dynamics by altering membrane composition in a slightly different way. One of the things that my colleagues did, again using these 3K synuclein cells, was to treat these cells with lots of different fatty acids, with different carbon chain lengths and different numbers of double bonds. And he found that by adding oleic acid, you increase alpha-synuclein aggregation, which is what we would expect based on the data that we've seen over time. And if you add the same carbon chain length, C18, with no double bonds, not only do you not have an increase in synuclein aggregation, but in fact this decreases. And the shorter the chain length, the greater the rescue. And so we asked, well, can we take a short chain fatty acid like C14 and rescue synuclein phenotypes? So here you see C18-1 increases synuclein aggregation, C14 decreases it. And when you co-treat with both, the C14 negates the negative impact of C18-1 alone. And indeed, this holds true as well for phosphorylated alpha-synuclein. And when we take the PD patient triplication neurons, treating them with C14 reduces phosphorylated alpha-synuclein. And as we suspect that what we're doing is changing fatty acid composition in membranes, we wanted to know if we were altering the interaction of synuclein with these membranes. And by sequential extraction assays, what we find out is we are. So we're increasing the amount of synuclein in the cytosol and decreasing the amount of synuclein at membranes. And to understand exactly how we remodel the lipidome, I'm just showing you one example here of phosphatidylcholine, where the chain length goes from longer to shorter as you go from left to right. And it turns out that when you treat C14 and rescue these PD-relevant phenotypes in these patient neurons, what you're doing is you're increasing shorter chain phospholipids and decreasing the longer chain phospholipids. And so overall, what I'm suggesting to you today is that it should be possible to correct lipid membrane dyshomeostasis and correct how synuclein interacts with those membranes, either by targeting lipid synthesis, lipid degradation, or with lipid treatments. And then finally, I want to thank my lab. They all look very happy here because we took a day out to a chocolate factory, but they do look this happy most days of the week. I've had some wonderful mentors, in particular Dennis Salko, and also some great collaborators, including Julian Pacheco, who does a lot of our fatty acid tracing. I want to thank all of the people who fund this work, and again to thank Parkinson's UK for being very supportive of me sharing my data here today. Thank you.
Okay. Thank you so much. I think we may have time for one or two very short questions before we close the session. And there's a question already in the back.
Hi, thank you so much. I'm really excited about this talk. It was really interesting because I work in mitochondrial disease and I'm really interested in fatty acid metabolism in particular. And so as you know, there is a lot of evidence for mitochondrial dysfunction in Parkinson's and in other related neurodegenerative diseases. Do you think there is a chance that the SCD and maybe even LIPE dysfunction that you're seeing could be a byproduct of the secondary mitochondrial dysfunction? Thank you.
When you say secondary, you mean, so you think the mitochondrial dysfunction could be causing this lipid dyshomeostasis?
Do you?
I think, I think a lot of my colleagues discuss this quite a bit, this idea that we don't know what the chicken and the egg are. So even if you think about lipids and synuclein, we know that synuclein alters the lipidome, and this is a problem. The reality is once you change the lipidome, you then change synuclein. So there's a lot of feedback loops involved, just like we saw with the GBA and synuclein earlier. We are also aware that there's a cascade of events. So you get changes in lipids, you get changes in calcium, you get changes in inflammation. What's not clear to us is the cascade that happens. I don't think it's a very clear cascade of one thing after the next. I think they're very clearly interwoven. And so when it comes to, mitochondrial biology, where lipids fit in that pathway is not entirely clear to me. I think there's also a very clear role for lipids that have some specificity to mitochondria, like cardiolipins, for example. They're not something that we've looked at in detail, but most definitely there's a connection between mitochondria and lipids. It's just where in that cascade it happens, we don't know, and it would be great to know.
Very good. Thank you. I think we've run out of time, Sarana, so thank you very much indeed for that. And I'd like to thank you and my co-chair, Esther, and Emil for presentations, and once again to Parkinson UK for supporting this, and to you all for listening to us, and for some excellent questions through the program. So thank you all very much, and enjoy the rest of the meeting.